Recording. Hi everyone. Welcome to the today's CAMBAM QLS talk by Melanie Prague. Dr. Prague is a permanent researcher at NRIA, University of Bordeaux in France, since 2016. She has led the statistics in immunology and translational medicine at INSERM at Bordeaux Population Health since July 2024. And she holds a PhD in biostatistics and public health from the University of Bordeaux as well. She obtained that in 2013. She completed 3 years of postdoctoral work at the Harvard School of Public Health, and that was in Boston, USA. Her research focuses on developing statistical methods for the treatment and prevention of infectious diseases. She develops both within host and between host models to accelerate development of treatments and vaccines, with her main funding centered around applications for HIV, Ebola, and other infectious diseases. Melanie, I'm looking forward to hearing your talk, and the microphone is yours.
Thank you very much for the introduction, and thank you very much also for the invitation, Emma and Alexander. I'm very happy to present today. So as you mentioned, I'm from University of Bordeaux, and I will discuss some of the work we lead in the teams that are mostly methodological. And in particular, we do work a lot with mechanistic models. So I will introduce what I mean by that later, but it's basically nonlinear mixed effect models. And I will try to introduce like 2 methods we've developed on how to integrate large to high dimension markers in those type of models. So of course, it's not something that I'm doing alone. So in Bordeaux, we are a big group working on vaccine development, and with so we are almost 100 people, I think, working on this topic, and maybe 20 of them are mainly dedicated to developing new methods in biostatistics. So we really have a focus on methods and on computational mathematics. So the work I'm presenting today, the 2 other PI who run this work are Rodolphe Thiebaud and Cécile Prost-Lima. And this is actually the work of our PhD students mostly. So Oriane Gabot, who now graduated, and Lisa, who is actually doing a co-tutel with Morgan Craig, who is at University of Montreal. So if you're really interested in this work, starting next September, Lisa will be in Montreal, so you will be able to meet her. So let's enter into the topic. So as I mentioned, I want to integrate high dimension data in a mechanistic model. And why? Just to start with some motivation, is because I want to do vaccine development, and I want to model the data I get from vaccination trials in order to answer questions such as how it works, how long, and then do some personalization, like how to predict the response in each individual, and also how to try to better find what are the optimal vaccination strategies. And usually what we used to have in the last 20 years were very, very small data, in the sense that we had longitudinal data. So we follow up multiple individuals in times. They are sparse and they are noisy, but they remained to be low dimension. Maybe we had the antibody response as well as some cellular population, but that's what all. But now more and more what we get is some large dimension data. So it could be proteomics or transcriptomics, like all the omics things. And in particular in this talk, I will be interested in gene expression, like transcriptomics, measured by your RNA-seq, so bulk RNA-seq. Okay. I want to introduce this type of data and to aggregate all those data that are collected longitudinally. And to do that, I will use what I called since the beginning mechanistic model. So this is a population approach of nonlinear mixed effect model, which is composed of 3 features. So if I think about the antibody response post-vaccination, you have a figure here. You can see the antibody response evolving a long time. And if I want to model this trajectory, what I can do is use a structural model, which will be a nonlinear equation. And the specificity of the work and why it's also very computationally demanding is that most of the time we will base everything on differential equation, and in particular ordinary differential equation. So for example, if I play with this toy example, if I want to understand like what could create this biphasic trajectory that I see here, then I will have like two population of cells, the short-lived antibody secreting cells and the long-lived antibody secreting cells. And they all, after vaccination, after a time of onset, they all just die at a certain rate, delta S and delta L. And those two types of population will also create at a certain rate some antibody that will also die at a clearance rate, which is delta AB. And the story I just told you can be written like with a differential equation, ordinary differential equation that is here, plus some initial equilibrium. And this will describe for a given set of parameters the trajectories that you see here. The thing is that when I think about the data I collect in real trials for vaccine trials, I actually have not only one trajectory, but I have a bunch of trajectory all collected from different individuals. And so to model that in mixed effect model, we will say that all the trajectory are actually from the same structural model. But the parameter of the model that I just described as being the rate of production and decay will actually be individual specific. Each individual will have a different value, and this will be described with what's called linear mixed effect model. So if I get back to my example, I will have some parameter of my model. So for example, here I took like the production rate of the antibodies from the short-lived antibody secreting cells. So this theta will be actually a population value, which is a value shared by everyone in the population, plus the potential fixed effect of covariates. So depending on the age, for example, I may have a higher or a lower, probably a lower, because we know that elderly have a lower antibody response. So I may have a lower response if I'm older. And some extra term here, which is just called a random effect, and which is here to be normally distributed in the population and to model really the residual heterogeneity that I may observe and that is not captured by the covariates that are deterministic and fixed in my model. So once I have that, if I think again about my data, what I have is actually data that are followed longitudinally, but not at every time point. It's only followed at certain time point, and it may be very sparse, as well as with some noise, as you can see here with those little waves. So it's not a perfect measurement of what's going on. So I need an observation model. And this observation model is first here to reflect the fact that I may have an error model. So we could think about a constant error model like a white noise. But on top of that, compared to the systems that I described, where I had like short-lived and long-lived cells and antibodies, what happened really frequently in the study we have is that we do not observe all the compartments, and we will call some of the compartments latent compartments because they are not observed. And in particular, if I think about these data that are here, I'm only observing the antibody response. And I can say that what I'm observing, so the antibody response, is a byproduct of what is predicted by my structural model with, on top of that, the statistical model with some error measurements that is constant. So this is what I call mechanistic model. It's nonlinear mixed effect model. And this is what we use to do some within-host modeling. So it could be immune response or virus dynamics. But this is what we use mostly as a way to model the longitudinal data we get. So first of all, one of the problem, which is not the one that I want to address today, but there is a bunch of work ongoing in my team with some students working on how to solve this inverse problem. But what I want to do is, if I'm given some data from a trials, as it is here, how can I solve the inverse problem and find all the parameters, so both the fixed effect, effects of covariates, the random effects, the variance of random effects, and the variance of error measurement, how I can recover all those parameters from the data. And here in this talk, I will assume that this is not the problem. And this is actually done with an SIEM algorithm as it is implemented in a software which is called Monolix, and which is an industrial software, but which is free for people working in academics. So, okay, so this is not the problem, but yet the question here is, this is really easy to run this type of modeling if you exactly know how to map what you observe with the compartment of your mechanistic model. And also if you have very few measurements, say, for example, only antibodies and some type of antibody secreting cells, because it could be measured. It's quite costly, but in some study we may have that. But now, if I'm also given, either at a single time point, say at baseline, or longitudinally, some gene expression, like I have no real system and no real idea how to link it with this. So I want to provide like new methodological tools to do that. And in this talk, I will talk about two different work that we were ongoing. So the first one will be the idea that if I have baseline transcriptomic, I can include it as covariate in my statistical model. And you will see what are the good places to do that. And if, on the contrary, I have longitudinal measurement of transcriptomic, how I can include it in the observation model. Okay, so let's start with the first part, which is using transcriptomic data as explanatory covariate of the dynamical behaviors that I see. So this work has been already published. There is a package which is on the CRAN, and there is a paper that has been published in January this year in Statistics in Medicine. So let's start with a little bit of motivation, why we will do that. So I want to associate and identify early biomarkers that are predictive of long-term response. And why it is a major thing in vaccine development is because most of the time when we give a vaccine, we have to wait, say, 6 months or a year to see if there is a persistent and long-lasting response. However, if we were able to find some early signals at maybe 3 hours post-vaccination, we would be very happy, because then we could do evaluation of vaccine candidates much more quickly and maybe do some optimization of those regimens and, yeah, to some extent, like reduce the cost. So the idea is that at vaccination pre-3 hours, sometimes we have some gene expression which is measured, and we want to know if what is measured here is actually a proxy of the long-term prediction that would be provided by a vaccine. So in order to understand a little bit more what it could mean, I would talk about the application that will come later. And in this application, so this is a clinical study of human response to vaccination against varicella zoster virus. And the design was the following. So we had longitudinal measurement of the antibody response, which is here at 7 time points. And we also had like the transcriptomic, so the gene expression measured at 3 hours post-vaccination. And so we had a total of 10,000 genes. And the big question is, among all those trajectories here that we see for the antibody response, we see that there is good responders, like probably the ones that are higher, and worse responders, the ones that have lower antibody response. And the question we want to address with the mechanistic model is there some information in these 10,000 genes that will explain who will become a good or a bad responder? Okay. So to address this type of question, there is a number of simple regression-based methods. There are other methods that are rank-based, and one also that is published in the team by Boris Bloom. But here the specificity is I don't want to just focus on one single endpoint, which is maybe what is the antibody response at 1 year. But I really want to know if there is an effect of my transcriptomic on the full trajectories using mechanistic model. So the idea to do that is to use covariate in the statistical model, and this has been already published, and there is a number of methods of there. So if you have some covariates that are low dimension, so say we just have the sex, the age, and the continent, there is a number of methods that will help you find to which parameter the covariate you have are related. So what we want to do is if we take back the statistical model, which is here, we want to know which parameter of the model, so which one of these arrows are null and which one are non-null and will have an effect. So which of those covariates have an effect on the parameters. So this, we had done that for Ebola virus, for example, in 2023. And if we think about how the methods that are out there, there is a lot. So you can test all the models possible. But the thing is that it's often not possible because, as I mentioned, everything is based on differential equations. So you need a solver. So it takes a lot of time. It's very computationally demanding. And then you have a range of methods, starting from the most exhaustive still, which are stepwise covariate modeling, and to others that are Samba, that are testing less models, so are probably less optimal, but in terms of computation time, they are still exploring the space and finding model with good properties in a reasonable amount of time. So here we will base our work on the Samba algorithm. And the question will be, now the Samba algorithm has shown to be working if you have like low dimension covariates. If now I'm adding my gene expression at 3 hours post-vaccination, and I also want to test how all of these genes are associated with the parameter of my dynamical model. So I will have a number here, which is very, very higher than 1,000. So it could be 10,000. How I can here just do the same selection as in Samba. So to address this problem, I will just, without really entering in the detail, try to explain how Samba is working. So you start with a model which is wrong, and iteratively looking at the residual of this wrong model, you will learn from the previous worse model in order to move toward a good model for the covariates. So for how all the covariates that you have out there are linked to the parameters. So the idea is that you start from a model, which is probably not the right one. So it could be a model without any covariates, so that there is no effect of covariates. Here you do a step of estimation. So as I mentioned at the very beginning, this is supposedly not the problem. So we know how to estimate the theta, and here it's done with the SIEM algorithm. And once you have estimated the theta, you can run MCMC procedure. So you can sample actually the posterior distribution of the individual parameter, knowing the data and the population parameter estimated. And we will take this simulation of the posterior distribution as a byproduct on which we will be able to run just like a simple linear regression. So basically what is costly here is really this step, because this is the estimation of the full nonlinear mixed effect model. So it may take a lot of time. Like I do have models that are running on large data sets that takes 12 hours just for one estimation. But here it comes to be a very, very simple problem, like linear model problem. And the idea of Samba is that we can do a down selection from this byproduct we got from the model, the posterior distribution of individual parameter. Then we can, for example, do a step AIC and select the model and select the covariates here that are linked with the sampled individual parameter and provide the lowest information criteria. So we do use the BICC, but you could use AIC or another criteria. And once you have a new proposition here, which is the best model for that, then you can plug it into a new model. And this new model will be estimated in the very time-consuming nonlinear mixed effect model estimation. So that is how Samba was working. And at first, we tried it with gene expression, but then it completely failed because STEP-IIC is not here to deal with large P, so a large number of covariates here. So what we did is just we transitioned the STEP-IIC to be a thresholded lasso procedure. So basically, we just transition this as being a lasso. So there is nothing very difficult here. It's really implemented just as in GLMNet. And this lasso procedure will actually help and will reduce the dimension of X and help dealing with the high dimension. So just in terms of simulation, so we did some simulation experiments. So in this simulation experiment that I will show you, but we did a bunch with different settings. So I will suppose that I have 200 correlated Gaussian covariates that you see here. And among those covariates, I will have 3 of them, so one that I called H, G1, and G2, that are associated with some of the parameters of my mechanistic model, so the production rates and the decay of antibodies. And I assume that I have 100 of individuals. And I will perform 100 replicates. And the question is, like running the tweaked lasso Samba, do I manage to recover the fact that I have those 3 associations and all the other genes, even the ones that are correlated, are actually not associated with the ongoing process described by the mechanistic model. So to assess that, we just define like the classical false discovery rates. So it's based on the fact that you will compare if a covariate is or is not in the generation model with the fact that it is or it is not selected in the final model after the estimation procedure. And we looked at 3 indicators, the false discovery rate. So we want that covariates that are not associated with the process are not selected. So we want it to tend to 0. And the false negative rate, in which you do not want to select an association if it was not in the generation process. So you want it to tend to 0 as well. And then you have the F1 score, which is just a balance between the two, and that is reported in this type of study in the literature. And you want the F1 score to be as close as possible to 100%. So if we look at the results here, so we just compare, so we know from the literature that the STEP-IIC Samba is faster and has similar performances as SCM. So now the question was to compare STEP-IIC Samba with the new developed Samba with the lasso procedure included. And so what you see here is that in terms of false discovery rate, there is a very high number of false discovery rate for the STEP-IIC, which is normal because STEP-IIC is not here to deal with high dimension, but it's way better with the lasso procedure. They have similar things in terms of false negative rate, and for the F1 score, it's also better for the lasso. And what was also interesting is that we could think that it would be worse in terms of computation time because the lasso has not real built-in properties to actually decrease the information criteria. But in fact, we see that because it requires less iteration in the Samba algorithm, it has like similar to better performances in terms of computation time. So now let's back to the application. for the application, so just to recall, we have these 35 adults, and we want to link the gene expression at so here it was at 1 day and not at 3 hours, and to link it with the different parameters of the mechanistic model. So first we did kind of a down selection. Just we went from 10,000 genes to a little less than 1,000, just by selecting only the protein coding genes that are on the pathways that are known to be associated with vaccination, just to reduce a little bit the size of the problem. We also tried other things, but like that was found as being good in terms of dimension reduction pre-processing. And then in terms of genes, so we run the lasso Samba procedure. So first of all, the initial Samba step IIC procedure just failed because it couldn't run. And for the lasso Samba, it provided results. And so we found that only 2 genes were associated with the production either by the short-lived or the long-lived of antibody secreting cells. And we kind of validated by cross-validation this result just to see if it was just a residual thing or if it was something more persistent. And we could see that there is kind of a signal that is, even after cross-validation, consistent over this data set. And so if we want to go a little bit deeper in terms of interpretation and also in terms of effect size, so here is there is a coloration. So what you see here is that so for KIFC1, it's associated with the production from long-lived antibody secreting cells. So it explains that it really splits better at the very end here with lower value of KIFC1 having higher value for antibody response for the expression. And so this, we found these genes, and it has been shown to be associated in other immune diseases. And in fact, it's really associated with the B cell development and also the stability of this development. So we could see, we don't want to overinterpret, but we could see why this gene could be a little bit interesting. And the other one was LEP, and it was mostly playing on the peak, so the production from short-lived. And LEP is also a gene that is a key player, in particular when you look at obesity and immune response to vaccination. It's been found as a gene that has a high impact. So we could at least make sense of what was going on and what was out of this study. So as a conclusion, definitively, so it's more efficient than the existing studies. So if you want to add covariates in your mechanistic model and it's high dimension, it's working better than others, I would say. However, there is still a very high computation time. And also, I would say that one of the perspective, which is also kind of a limitation right now, is that I think it's a little bit naive to just interpret like one gene, because we do know like all the genes are really working into pathways or gene sets, and they need to be grouped and they have like joint effects. So I don't want to overinterpret like why we found one gene or the other, but I rather say that one of the perspectives would be to work on more structures in extension of this to actually structure the correlation we have between the genes. But still, if you're interested in that, it's out there and it's available on the CRAN for those who are using R. Okay, so that was for the first part of the talk, and the second part of the talk is still handling this type of high dimension transcriptomic data. But instead of assuming that we have only one time point at baseline, we assume that we have like longitudinal measurement of it, and it requires other methods. And The motivation here is a little bit different. It's not really to find one surrogate marker, but it's more to be able to have a good digital twin of the immune response. So we know that vaccination is super complicated. We still probably do not understand everything. We would like to try to understand this response with models that are super big and that are kind of modeling a lot of different type of cells and of processes. But we end up like having to analyze it with very, very small model. And in particular, because of the problem of identifiability that I did not mention much, but which is one of the biggest problems suffered by mechanistic model, which is that, I mean, even with a simple model like it is here, if I have a lot of latent compartment and I'm assuming that I'm just measuring one, then it's very difficult or impossible to actually recover the value of all the parameters of my dynamical model. So we need to add information into this model. And this is where the idea of having longitudinal transcriptomic information is very appealing. And why is that? So there is a recent study, again, it's beginning of January 2026, in which it's been shown. So this was in a patient who had COVID. So basically what was measured was prick test transcriptomics. So the prick test has advantage to be feasible almost every day or very longitudinally, so it will not be very sparse data. And it's been compared with the transcriptomic information that is drawn from tempus, so like full whole blood analysis. And so what was found is that first there is a good concordance between the two, so we can use prick test to actually record transcriptomic data. And in the blood sample, it was also measured the cell abundances, and this can be done with other statistical approach, which are mainly based on deconvolution approaches. And so basically, the deconvolution of the transcriptomic data giving prediction of cell abundances is very consistent with what has been measured in this study with flux cytometry. So it means that probably in the transcriptomic data, there is some type of information about cell abundances that we will be able to include in our mechanistic model. So for this reason, we developed a method which is different from the other. And we based everything on one motivating study, which has been published in Rinchai during the COVID epidemics. So it was only 15 individuals that were vaccinated by the Pfizer vaccine, and they were followed up at 6 time points for the antibody response. So here you can see the data. And just because we were not modeling like for the varicella zoster virus, only the long-term response, but also the establishment of the response, we had to add a compartment here, which is like the triggering of the creation of short-lived and long-lived antibody-secreting cells. And so it adds one compartment to my model. And what we have is that if we only try to use this data and try to fit this model with the available tool, we find that we will have to fix so many of these parameters because the model is actually not identifiable. It's too complicated for these data that are really sparse, and if we only observe one part of the gene. So what's good is that in this study, what was measured as well was the gene expression, so longitudinal gene expression every day through prick test as well, up to 14 days after prime and then up to 35 days after boost. And so just in order to reduce the dimension here again, so we went from more than 6,000 genes and we grouped them into gene sets. So we ended up having like 34 information a long time about the different gene sets according to Chosabel. And so we wanted to associate those gene sets with the dynamics of the latent compartment that were here. So to do that, the idea is to use the longitudinal gene expression as an observation of the latent compartment. So before, I mentioned at the very beginning when I introduced the mechanistic model that here the antibodies will be directly observed through the byproduct of my ODE, which is reflecting the antibodies. Here for the gene, it's a little bit more complicated. So I know that what I observe in blood is not what is happening in the germinal center. So it has to be something which is very transient. So we associated it with the latent compartment of the antigen and the dynamics of the antigen, saying that the antigen will trigger directly a response in the blood, and this response has to be reflected in the gene. And so in the observation model, I will say that all the genes that are measured here will be like a base expression of each gene or gene set, plus the reflection of the fact that we have the latent compartment AG, which is moving over time, plus some error. And the question here, and why I will have to build a procedure for estimation, is that I cannot estimate all the alpha plus all the parameter of the mechanistic model jointly because it would be too many parameters. So I have to perform some regularization here to see which genes are actually associated with my latent compartment that I call the antigen here, and which genes are actually not associated with that. And so to do that, we just implemented a two-step method, which is again based on a lasso procedure. And so the idea is for a given lambda for the lasso, we will have a penalized likelihood, and we will do a two-step algorithm. A first one in which we assume that the parameter of the mechanistic model are known, but we regularize the alpha 1 using a thresholded lasso. And another step that we assume now the alpha 1 known, so we know which parameter, which genes are associated with my latent compartment. And I use the SIEM algorithm just to estimate all the parameters of my mechanistic model. And I run onto those iterations until there is a stabilization of the algorithm. So, of course, because it's a lasso procedure, there is a problem of the selection of the lambda, so the penalty in my penalized regression. So I could do that for each lambda. And so we had to define a grid, and so we found this. So this has been described in this paper, and this is a way to define a grid that is sparse enough so that we don't have to run too many estimation. And then at the end of the day, we can run the procedure that I just described before for different value of lambda. And then it provides you with this regularization plot in which you see that for each genes, it's fastly going to zero for some, and for others, you have to have a penalization that is quite high so that it's actually regularized and we find no signal. So once we have this regularization plot, we still have to find where to cut. And the thing that we used here was, again, the information criteria, assuming that the model that will be both the best in terms of adjustment and in terms of penalization, because it has a lot of parameter, will be the best one. So for each lambda, we can compute for the final selected model what is the information criteria. And for this information criteria, select the model and the regularization parameter that is associated with the lowest information criteria. And at the end of the day, that will provide you with a model fitted in which you have only some genes that are associated with your latent process. So again, we run some simulation. So just to give you an idea in this simulation, this is a noised gene, and here this is a gene that is not noised. So we can see a signal, but this is not always a signal that you can actually see just by eyes, I would say. And I don't want to enter too much into detail, but I think that what's important is that even in this simulation, if I just take the antibody, and I try to do the estimation in the simulation, then I cannot estimate from only the antibody data the model. It will be non-identifiable. I will not reach convergence and I will not believe the results. But if you add the procedure and all the gene expression, then you will end up having an identifiable model. So all the estimation will run well. And as well as if you look at all the criteria that are very mainstream, like the bias, relative bias, and the coverage rate, then you can see that they do achieve like a small bias and coverage rates that are quite similar to the nominal expected level of 95%. So we do manage to reach the estimation. And on top of that, if you see, because it's simulation, so we know what are the genes that are associated with the latent compartment. We do manage to have a very good recovery of that with a very few false negative, if it's not none. And for the false positive rate, we see also that there are very, very few genes that are set to be associated with the latent compartment, whereas it was not the case for the simulation. So this was just for the simulation. So we run that, and we are still running like few extra simulation in which the ratio for signal to noise is a little bit less in our favor. But if we move to the application again, so those are the 34 gene sets. So the gene sets are really coding for different functions. So it could be monocytes, B cells, cell adhesion, cell death, etc., etc. And we had like those 34 gene sets measuring each individual very longitudinally, like with a very good sampling. And we assumed that we wanted to know if each of these 34 genes were or were not associated with the antigen dynamics. And on top of that, we have, of course, the antibodies response that were measured directly. So what we did is that we ran the algorithm, and the algorithm is quite long to run. It's like more or less 1 day for one run, so it's like quite a lot. But what happened is that we looked at the regularization plot. So we can see that the regularization plot is a little bit less clean. And if we look at the minimal BICC, it's found for a penalty term that is quite small. But yet it's regularized for some genes, not all of them, but some genes. And if we look onto which genes, so at the end of the day, it will select 11 genes over the gene sets, gene pathway, over the 34. So we assume that only 11 of these pathways are actually related to the vaccination process. So among them, we can find like a lot of different ones, so monocyte, cell adhesion, cell death, cellular respiration, etc., etc. But then if we rank them by the magnitude of the alpha 1, assuming that the one that have the strongest alpha 1 are probably the one that are mostly associated with this response, then we end up finding at the top 4 the inflammation, the type 1 interferon, and neutrophils. And those 4 pathways are really pathways that have been shown in system biology with approaches that are completely different. They have been shown to be really related with the vaccination process. So we managed to regularize to select only a certain number of gene sets that are associated with a response, and the one that are more strongly associated with the response are as well the one that we would expect to find. So as a conclusion, I would say that the advantage of this method is, of course, it's the only one that really allows a simultaneous selection and estimation of the gene latent compartment association. So to the extent of my knowledge in the framework on nonlinear mixed effect models, there is no much method out there. And what is the take-home message? The take-home message is, first, we managed to do a regularization, and it seems that both in simulation but also for the real application in some nice data, we managed to identify more complex model, and that would open the door to building like more complex mechanistic model of the immune response on data that are more complex, in order to have what we could call digital twin of immune response that would be more meaningful and less of a toy example and a little bit more going on to the processes. So of course there is tons of limitations that I'm happy to discuss. So right now I would say that one of the strong limitations is computational cost, and especially here because I talk about large dimension, because we only have like 34 gene sets. If we wanted to work at a gene level, I think like this procedure so far will completely fail. So we will have to see how to deal with that. And the thing that is mostly a limitation is that it's only taken into account right now, as it is on the CRAN, only one latent compartment. So it means that right now we assume that there is no S and L, for example, here, and the gene is only reflecting one latent compartment. But this is probably not true because it's bulk RNA-seq, it's not single cell, and it's in blood. So they're actually a mixture of multiple cells, and it could be like a mixture of what is the response of antibody secreting cells, short-lived, long-lived, and other cells. And this is a mix of all these cells that is actually producing the gene expression that is shown. So actually we are currently, and this is what Lisa Crépin is doing right now, extending the algorithm so that now it really reflects this biology, and say that gene are the reflection of maybe all the compartment of the model if there is no further information available from biologists. If you're interested in that, there is already like a lot out. There is a vignette that you can find on the CRAN. There is a package which is already on the CRAN. And if you email me, I can already share a draft of the simulation and so on. And it will be very soon available on arXiv, and hopefully sometime in a journal. As a conclusion and perspective, I would say that first it's out there, and of course there is a lot of limitation, as I discussed before, but I think those two packages are answering different questions. I try to motivate in each of the two introductions. And those they are out there, and we are also looking for other relevant application, because we do have some in immune vaccination and in virus dynamics. But I do believe that in particular the lasso samba would be very useful in pharmacogenomics, and we couldn't have access to some data, and we didn't really look for that. So that's for the first part. And for the second part, I think there is a very good application in cancer research as well. Just because deconvolution at the very beginning is some methods that are really, really well developed for cancer research and for solid tumors. And so I believe that here, because those deconvolution approaches are very well fitted for this type of methods, there is no reason why, if you have a mechanistic model of tumor growth, say, it couldn't be related with gene expression that is measured as well. But this is not the area of research we explore in my team. But if you are interested in that, I would be very happy to discuss. And yeah, and so with that, I just want to thank my collaborators, the Vaccine Research Institute, which is funding a lot of our research, and all other funding agencies and colleagues, and all my team, which is awesome. And if you want to contact me as well, either for this project or because you're a student and interested in working on some related projects, that would be amazing as well. You have my email here. And thank you very much for the invitation and for listening. I'm happy to take questions.
Thanks very much, Melanie, for an interesting talk. For questions, you can either unmute yourself and ask your questions or write your question in the chat, and I'll happy to read it for Melanie. So please go ahead. Okay, while we're waiting, maybe I'll ask my question. It's more related to first part and actually related to the second part as well. You're basically at least in your mathematical model, like mechanistic model, you have these two compartments. You have the short-lived and long-lived cell populations, and it's kind of a prior in your in your algorithm. You're assuming that you have these two subpopulations to produce the biphasic response and in your antibody response. Why
Yeah.
do you need to do this? I mean, you could basically have one compartment model, essentially. They're talking about one cell population and let essentially the algorithm to determine what these parameters are, the degradation rate of all these antibodies are. You know what I mean? Like, I mean, the thing is, yeah,
Yeah. So you mean that here there is a strong prior that this is a biological structure and
you're assuming a structure, which is a prior you're assuming within the model.
mhm.
Um, why do you need that step instead of just keeping it?
Parametric or just
Yeah, exactly.
yeah, so I think so. This is a good question, very good question. Thank you. So I think like using a mechanistic model in that sense that they are delineated with a structural model that is based on biology and that is based on biological assumption is really a strong position, I would say. So we have a bunch of paper in which we kind of compared it with some spline approaches, so that are like highly non-parametric and they can model whatever. And I think that why I want to use that is really a standpoint, is because we do work with biologists that have very strong view on what is the ongoing process and how cells are interacting with each other. And the fact of using this type of very structured model with a prior specification of the interaction between the latent compartment introduce a lot of prior and a lot of information. And in particular, what we showed in one paper is that if you, instead of doing that, you use a spline, then the spline will never be able to do some prediction outside of the range of time points in which you learned on your data set. Whereas those type of mechanistic models, because they are encoding biological knowledge, they may have steady states, and they may help in exploring better and further outside of the range and the time frame of the study, as well as with other counterfactual scenarios, I would say, like what if I change some value of covariates to some other value and things like
Mhm.
that. And with spline, I would be less trustful of the results, mainly because it's not encoding any medical biological knowledge.
Mhm. Mhm.
But it doesn't mean that in some applications, splines are not better, right? But
Yeah, I see. All right. I'll give people a chance to ask their questions. There's one on the chat. This is from Ryan. It says, curious, how was the gene set value computed? Was it just
yeah.
averaged, or yeah, you can read it. Go ahead.
Yeah, I read it. So this is a good question, and this is something that we had to make some choice on, and maybe it's not the best choice. So we took the Chusabel pathways. We grouped our genes from the study on that, and then from each individual, we just averaged it out over all the genes in this pathway. And you're right that if two genes are completely uncorrelated, then it will average out to zero. But we found that it has been done in other studies as well. And I mean, this was more like of a proof of concept right now rather than like really trying to have definite answers to build the digital twin. So far we just work with that. And I agree, it's not perfectly satisfactory. Hopefully, we are still resuming summarizing some good information because we find pathways that are meaningful. But I definitively agree that if we could go to a layer that is more granular, instead of gene set, we would move to gene. That would be better. It's just that right now it's not possible in terms of computation time, and I have no good idea to deal with that yet. Thanks.
Any other question?
Yeah, I had a very quick, very high-level question. I was wondering, I mean, because this is, again, this may be completely wrong, so please just tell me. Because it's fundamentally like observational data, there's standard problems from regression, like missing data, not unmeasured data, confounders, and all these should pop up here as well, right? And then how do you deal with those when you're trying to draw inferences, like, for example, especially unmeasured variables?
Yeah. So missing data, we deal with it. I mean, we should be quite robust to missing data to some extent because we are having a likelihood-based approach. So we are missing to MAR. We are robust to missing at random data, and we assume that the missingness we get is
Sorry,
missing at
maybe I
random.
didn't explain it clearly. I actually meant unmeasured variables, like covariates that are not measured.
Okay, and so that's a good question as well, like for causality. So we have a paper that was out in 2016 in biometrics, in which we tried to analyze data from a cohort study in which we had informative treatment initiation. So this is this type of confounding, for example, when the status of the patient is both predicting the outcome and the treatment assignment. And we showed that because mechanistic model are encoding the time in a differential equation, it's like a structural model. So time is only going one direction. So it's still a good tool to address question, even in the context of missing confounders. I mean, you still have some causal properties in this model that allow you to some extent, under some strong assumption, analyze data. But where you're perfectly right is that still it will be very problem specific, and the result will be just as good as the specification of your model at the very beginning. If you have like a very bad model with data in which you didn't measure too many compartments, then probably you will have rubbish out as well. So,
but is it possibly fair to say that if the characteristics of the population don't change too much, your prediction should still be okay? If the
yeah, that's the bet, at least. Yeah.
Thank you.
Yeah.
Any other question? Okay, so thanks again, Melanie, for your talk and for giving us some really interesting background on this topic. So it was really interesting. This is all. Thanks for everyone for attending, and we look forward to seeing you in next week's talk.
And just a note, next week David will be here in person.
In person, yes, yeah.
yeah.
Yeah, we invited the speaker next week to give the talk in person from the University of Michigan. So please come in person as well. Okay.
Thanks, Melanie.
Take care, Melanie. Bye.
Bye. Thank
Bye.
you.